כמו בכל תחום במדע, תחום תוסס ומתקדם, כמו שהודגם גם בהרצאה הראשונה על ידי פרופסור יוסי שלזינגר, יש התפתחויות אדירות בתחום. מה שהיה לפני 40 שנה, כמו שאמרה פעם גולדה מאיר, מה שהיה הוא לא יהיה, והדברים מתכוונים בקצב מדהים לפעמים, ומצאנו לנו לכבוד להזמין הנה חלוץ בדור הנוכחי של חזית המדע בתחום שימוש בכלים אופטיים לחקר המוח, פרופסור יזהר ממכון ויצמן, שכמה שאני זוכר גם זכה להכיר את עמירם, ועופר ידבר על Beyond Optical Imaging, Writing Activity into the Living Brain. עופר, בבקשה.
תודה רבה, ידין, תודה לכולם. זה כבוד גדול להיות כאן. כמו שידין ציין, החפיפה שלי עם עמירם במחלקה לנוירוביולוגיה הייתה מאוד מינימלית של כמה שנים, אבל בעצם כמי שגדל במזכרת בתיה בשכנות כמעט לרינה הילדסהיים ומשפחת הילדסהיים, אני סך הכל שמעתי על המחקר שנעשה אצל עמירם הרבה שנים לפני שבכלל התחלתי לחשוב על ללמוד חקר המוח, ובעצם דרך רינה וז'אן והילדים הכרתי עוד כילד את המעבדה של עמירם. כשהגעתי למחלקה אז דווקא יצא לי הרבה לדבר עם עמירם, כי הרבה מהדברים שאנחנו עושים, כמו שתכף תראו, נוגעים לתחומים שהוא עסק בהם, ואפילו קיבלתי ממנו עצות תמיד מאוד לעניין וחכמות על איך להתמודד, למשל, עם כל מיני קשיים טכניים שהיו לנו בדברים שניסינו לפתח. אז זה כבוד גדול להיות כאן ולדבר באירוע הזה. אני גם רוצה להגיד שבעצם אני עומד כאן לפניכם, אבל אני בעצם מייצג קבוצה די גדולה של האנשים. במרכז אתם רואים את הקבוצה שלי באחת האיטרציות שלה, וגם את אנשים שעבדתי איתם לאורך השנים, שעזרו בעצם וגם הנחו אותי, למשל קרל, שהיה המנחה שלי בפוסט-דוקטורט, לפתח חלק מהכלים והטכניקות שאני אספר עליהם, וגם בעצם להזין את הרעיונות לדברים שקורים במעבדה, שאני אדבר עליהם בחלק השני של ההרצאה. אוקיי. אז אני חושב שאני לא צריך לספר לכם על כמה המוח הוא מדהים, ואני בטוח שכולכם הייתם במקום הזה שבו אתם מסתכלים על הדברים שהילדים שלכם מצליחים ללמוד ולבצע, ותוהים איך המוח או פעילות של נוירונים במוח יכולה להביא לדברים המופלאים האלה. אנחנו עובדים במעבדה בעכברים שיכולים גם ללמוד דברים שהם יחסית מופלאים, אבל כמובן שאין לזה אח ורע במוח האנושי או בדברים שהמוח האנושי יודע לעשות. אני מקווה שחלק מהדברים שאנחנו עושים יעזרו לנו להבין קצת את הדברים האלה שקורים במוח האנושי. I'll switch to English, sorry. So we're interested in understanding how neurons in our brain encode the outside world, like the beautiful talk that you just heard about from Hamutal, and also how they drive specific behaviors, how changes in the connections between neurons lead to the formation of memories and the erasure of memories, and how these things change in animal models of psychiatric and neurological disorders. And I hope that understanding something about the links between neural activity and behavior can help us understand what happens when things break down in brain disorders. So these are the two primary questions that drive the work in the lab. My lab is actually two separate labs. One of them is engaged in neural circuit research. We study, as I said, mice. We work on areas in the mouse brain that encode behavior, that drive reward-associated behavior, that lead mice to develop specific behaviors that allow them to obtain rewards and to learn from their experience. And as you already heard, recording the activity of neurons while recording behavior, like the beautiful examples that Hamutal showed, actually help us understand something about the correlations between them and think about how particular activity patterns in the brain might be linked with behavior. The trouble is that while we can learn a lot from understanding correlations between brain activity and behavior, this leaves unanswered questions about causality. So the causal effect of whether these three neurons are active while this mouse is touching the screen to obtain a reward doesn't tell us that these three neurons are actually responsible for that behavior. For example, figure ground segregation might be driven by top-down projections from higher visual areas, but until we're able to shut down those top-down projections and see the effect, first of all, on the representation of figure ground segregation and on behavior, we won't be able to really know that this is really the circuit mechanism for that behavior. And this is the reason that the other part of my lab is engaged in developing new neurotechnologies that allow us to make slightly more confident claims about the role of specific neurons, the causal role of the activity of neurons in behavior. And these techniques collectively have been referred to as optogenetics. It's a new neurotechnology that's been around for the last 15 years. It's not so new anymore, but there are new and improved tools coming out all the time. And I'd like to share with you a little bit about how this actually works and what we've done to allow a higher resolution study of the causal links between neural activity and behavior. So the main challenge, as you probably all know because you all think about the brain to some extent, is that the brain is extremely complex. What you see here is a picture of a tiny piece of a mouse cortex. The neurons are stained using a genetic technique in which each neuron receives a different color based on the specific genetic profile of genes that it expresses. It's called the rainbow technique, not from our lab, but just to demonstrate the idea that each one of these neurons might be encoding a completely different function. And this is, even though, for example, sensory cortex that you just heard about is very organized, associative cortical areas, such as the prefrontal cortex that we're interested in, are much less organized, and the different functions that neurons perform are not really divided up into different places. So there's no association between place and function in these higher order brain areas. And for this, in order to ask questions of causality in this kind of complex circuit, you need to have a technique that allows you to single out particular types of neurons, like these green neurons here, and selectively manipulate their activity, either activate them or inactivate them, and then study in the living brain how that specific manipulation actually changes something that the brain does. It could be how these neurons change the activity of their neighbors, which would tell us something about the connections between the neurons, and it could be how the activity of these neurons actually changes the animal's behavior that you're observing at the same time. So this is kind of the challenge. And the challenge is not new. It was actually set forth by Francis Crick, which probably many of you have heard about. He's a pioneer of molecular biology. But he's, after he finished that little problem of solving how DNA works, he became very interested in the brain, and he wrote a lot about the brain, eventually moving to California and working on questions related to brain function. And he wrote in the Scientific American article in 1979 that the major challenge facing neuroscience is the need to control one cell type while leaving the others unaltered. And this challenge that Francis Crick set up sent many people at the time. I was 4 years old at the time. I did not read this article, but Amiram actually read this article, as he told me later when we met when I joined the department, and him and many others have started to try and match this challenge and find ways by which this can be done. And when I first met with Amiram in his office at the Weizmann Institute, he told me, you know, I was actually the first one who almost did optogenetics, and this was in 1983. We published it in Science. This was a paper that he published together with Ira Farber, where they developed a technique which kind of typically of Amiram's creative mind, used the tools that he was already familiar with, so the membrane binding fluorescent probes with laser light, in order to stimulate neurons in the brain. They did it in various different organisms. This is a demonstration of the approach. A microbeam laser basically stimulates one neuron while another neuron is being recorded, and if these two neurons are connected, then that stimulation of one neuron can lead to a synaptic response in another neuron and will tell us something about the connections that exist in this particular circuit. So you can see these are simple organisms. It's a relatively simple technique. The basic idea is that you bathe the neurons in a dye. In this case, I think this was RH500. The dye makes the membrane sensitive to light, and then you come with a microbeam laser and you kind of tickle the neuron with light, which leads it to fire an action potential. This is the technique. It was revolutionary at the time, which is why it got published. And even in that paper, they noted that this can be very effective in simple circuits, but in more complicated circuits, or even in multi-layered circuits where neurons are sitting on top of each other, like we know the mammalian brain definitely is, there would be a problem where if the laser penetrates several layers of neurons, it will stimulate all of them. And this is because the dye is not specific to a subset of neurons. Okay, so this was a very clever technique. It was useful in simple circuits, but Amiram knew, and they noted this in the paper, that it doesn't fully match the challenge of leaving the other neurons unaltered. As you might know, Francis Crick was responsible for the central dogma of molecular biology, which basically states that proteins are encoded by DNA, which through RNA get translated to proteins. Unknowingly, perhaps, Francis Crick actually set up the creation of optogenetics, which maybe more fully answers the challenge that he wrote in 1979. And I'll tell you basically how the knowledge of how DNA makes proteins actually can lead to development of a new technology for stimulating neurons. So optogenetics is a very simple technique, basically. All basically it needs is insertion of a certain DNA into a subpopulation of the neurons, those neurons that we want to manipulate, which makes them sensitive to light selectively. Okay, now we have some of these neurons, these are the green neurons here, have been targeted with a genetic tool to make them light sensitive, and then we can shine light non-selectively over the entire circuit, and this would cause the neurons that are targeted genetically to express a light-sensitive protein to fire an action potential. That's basically the idea behind optogenetics, and this was done in 2005 by Ed Boyden and Carl Dijsselroth at Stanford, where I did my postdoc. The idea, or how it works, if you might be wondering, is that it's based on a very simple observation, which is that many microorganisms living around us in nature actually have light-sensitive mechanisms. These algae called Chlamydomonas reinhardti, you can see them here under a microscope. They're unicellular algae. You can't see them with the naked eye, but under a microscope, they look like this. In the dark, they move around because they have these flagella, but they move around in a random kind of Brownian motion, as you can see here. But let's look at what happens when these algae are illuminated with light. So if we shine light on these algae from the side, they make a photophobic reaction. They don't like strong blue light, and therefore they move away from the light. And they can do this because they have a certain protein called chanorodopsin in a part of the algae called the eye spot. And this protein is a very unique protein. It's an ion channel, but it's also a photosensor. Okay, it contains the retinal chromophore, a very similar chromophore to the one that you have in your retina and your photoreceptors, which allows you to see the world. So this algae uses this retinal protein in order to allow it to escape from strong blue light. Now, how is this related to Francis Crick in molecular biology? Because this protein is encoded by a DNA, okay? And if there's a DNA in this algae that encodes this protein, we can take this piece of DNA, we can put it under the regulation of a certain regulatory element or a promoter. We can package it into a viral vector. In our case, this is an AAV viral vector. AAV vectors are now a very hot topic in medicine because they've been used as gene therapy vectors. There are many clinical trials now using AAV vectors to treat human disease. And these vectors are relatively safe. They don't cause any disease. And we engineer them to contain only this gene under this promoter so that if we inject them into the brain, they would express in subsets of neurons. And the identity of the subset of neurons that would express these genes is determined by the regulator that we choose. So we can play with this like Lego, basically replace the regulator with another regulator, which will allow us to express in other populations of neurons. So this is kind of like a very versatile technique using light-sensitive proteins to sensitize neurons to light. And once this neuron expresses the channelrhodopsin protein, which it doesn't express naturally, it expresses it because we introduced it using our viral vector, we can deliver light now to this neuron, and it will respond with action potentials, which is what neurons naturally do when they're activated by their neighbors. Even more than that, we can play with the type of protein that we express. We can, instead of the channelrhodopsin, which is a positive ion channel, we can express another protein which conducts negatively charged ions. And if we go through the whole process again and put this protein, this negative ion conductor, into the neurons that we're interested in studying, it will now allow us to suppress their activity. So this is a bidirectional technique that allows us either to excite the neurons with blue light if they express channelrhodopsin, or inhibit them with yellow light if they express a protein called halorhodopsin. And this is the basic principle behind optogenetics. And you might be asking yourselves, and this is all nice, and these are might be neurons sitting in a dish, but does this actually work in the living brain, which we would actually need it to do if we're going to study behavior with it? So the first experiment done to actually ask this question was done by Viviana Gradinaro at Stanford, my colleague for many years, and she took mice that were engineered to express channelrhodopsin in the motor cortex, in excitatory neurons in the motor cortex. She placed a fiber optic cable above the motor cortex of this mouse, and you can look at the mouse's behavior and see if there's any change in its behavior when the light switches on. So this mouse is now running around in circles because the left side of its motor cortex is activated with channelrhodopsin, which indicated that even in the living mammalian brain, this protein that comes from algae is capable of causing neurons to fire action potentials and drive behavior when it's illuminated with blue light. So our lab is using this tool among many other optogenetic tools that have been developed since then in order to understand how patterns of activity in neurons drive behavior. We're particularly interested in understanding social behavior in animals, and I'll show you a little bit about that. But we're also interested in pushing the technology further. And as you can imagine, neurons don't just all fire action potential at once. They have very intricate activity patterns, which you might have seen in the first video of neural activity that I showed you. And bathing the entire circuit in light basically means that all of these neurons are going to fire action potentials at the same time. And we're going to be inducing very kind of artificial activity patterns. Now, a really exciting development that's been happening in the last few years is the introduction of two-photon technologies in order to further refine our ability to drive specific activity patterns in neurons instead of just triggering their activity all at once. And I'll show just a couple of slides on this. So we've been collaborating with Dan Owen at the physics department, actually chemistry now in Weizmann Institute, and Valentina Emiliani, who is at the Institut de la Vision in Paris, both physicists. So Dan developed a technique called temporal focusing and combined it with holography. And what this allows us to do now is to, instead of deliver blue light to activate all of the neurons in the tissue at once, actually make little holograms of two-photon illumination. This is infrared laser illumination, which also allows us to penetrate deeper into the brain. And using these techniques, we could actually place little patches of light in 3 dimensions in the tissue and actually individually stimulate each one of the neurons in the tissue instead of just bathing the entire tissue in blue light and stimulating all of them at once. So this allows us to do several things. It allows us to regenerate more natural activity patterns with light, with this 2-photon stimulation technique. But it also allows us to ask questions about connectivity, because now we can look at this neural circuit and stimulate each one of these neurons separately and ask how does that neuron trigger activity in other neurons around it, and essentially similar to the experiment that Amiram was doing in the frogs and mollusks in 1983, but now with better kind of spatiotemporal resolution. This is a collaboration with Tommaso Fellin in the Institute of Technology in Italy, in Genova. And what you can see here is that apart from stimulating the neurons with light, we can actually read out now their activity in what we call all-optical optogenetics. And these neurons are now not just expressing a channel rhodopsin, they're also expressing a sensor for neural activity. This is a calcium sensor, and you can see that the calcium sensor trace actually goes up every time we shine these holograms to stimulate the neuron with light. You can see that calcium activity goes up, as well as spiking activity that is simultaneously recorded. This is what the tissue looks like. It's red because the calcium sensor is red, and the green neurons are the neurons expressing a high potency channel rhodopsin that we engineered to express in excitatory neurons in this area. And you can see here that using this technique, you can actually stimulate multiple neurons, multiple single neurons now at once in the tissue. So just a little bit about the scientific questions that we're asking. So you've heard a lot about cortex. We looked at columns and representations in large-scale cortical tissue, but we also know that the cortex is actually an extremely complex tissue which contains dozens of types of neurons. And probably the simplest distinction of cortical neurons is to excitatory and inhibitory. Excitation and inhibition in the cortex is tightly balanced. These neurons, these green neurons, would be excitatory neurons. These red neurons would be inhibitory neurons. And when one of these neurons fires, it will activate other neurons around it. When one of these inhibitory neurons fires, it will actually dampen neural activity around it. And as you can imagine, if we lose the activity of these inhibitory neurons, this can be detrimental to how the cortex works. So the disruption of balance between excitation and inhibition has been hypothesized to lead to multiple types of brain disorders, including most prominently epilepsy, but also autism and schizophrenia. And we're interested in understanding how the disruption of this balance actually leads to dysfunction related to these disorders. This theory of excitation-inhibition balance was proposed 20 years ago by Rubinstein and Merznick, and we were interested in trying to causally interrogate this question of whether experimentally disrupting the excitation-inhibition balance would actually lead in animals to changes that are reminiscent of the changes that happen in the human, in these human disorders. So in order to do that, we looked at the behavior, the social behavior of mice that we engineered to express a unique type of channel rhodopsin, either in excitatory or in inhibitory neurons in their prefrontal cortex. And we looked at the details of how the mice socially interact with each other. I'm not sure if you know this, but mice are extremely sociable animals. They live in social groups, and they have a very defined social hierarchy. And we asked whether changing the excitation-inhibition balance by disrupting the activity of, or elevating the activity of the excitatory neurons in the cortex would actually disturb the activity, the behavioral activity of these mice. We can do this repeatedly, and we can check the behavior of the animals both when the light is on and when the light is off, so that we can always compare light on and light off condition. And when we looked at the behavior of the mice, we found that in the control mice, as you can see, they typically spend more time interacting with a new social partner than with an inanimate object, which is what you see here, whether we deliver light or not. And so the native brain doesn't really care about blue light because the neurons are not sensitive to light. When we increased excitation optogenetically, we found that the mice that expressed the channel rhodopsin in their excitatory neurons actually lost their social preference which indicates that the excitation in the prefrontal cortex, or elevated excitation in the prefrontal cortex, actually contributes to social dysfunction in mice. Now, when you study social behavior, you're obviously studying not just one brain region. As you can imagine, there are many brain regions that contribute to social behavior. It's a very complex behavior. And the social network, or the network of brain regions that contributes to social behavior, has been at least partially mapped out. And we can today try to ask questions about how these circuits interact with each other and how each one of these nodes in the circuit actually contributes to a particular aspect of social behavior. And this is a long-term goal of the lab. We're interested in understanding how changes in the activity of the connections between each one of these brain regions. Here you can see the prefrontal cortex and amygdala and nucleus accumbens. All of these areas have been suggested to be involved in social behaviors, so is the links between them. But asking questions about the links between brain regions is extremely complicated. As anybody here who's done this kind of experiment before, what you see here is a DTI map of the human brain. You can see that the brain, these are basically the wires or the neural pathways connecting brain regions. So essentially what we're trying to do is ask what specific parts or specific connections, specific wires in this diagram actually does in the context of the living brain. So in order to ask that kind of experiment, for example, how this pathway specifically contributes to behavior, we need tools that allow us to suppress temporarily the activity of this pathway and then ask how the behavior of the animal changes. And each one of these wires is composed basically of axons connecting one neuron to another neuron in another region, and the ideal method to allow us to ask this kind of question is to illuminate specifically this axon and suppress temporarily the activity of these synapses that connect these two neurons. Matthias Mann, who was a PhD student in my lab, tried to approach this with the conventional optogenetic tools that I've just been telling you about. We found that while channel rhodopsin is extremely useful for excitation, it's actually not really useful for silencing, as you might expect. The tools that were effective for silencing at the neuronal soma were actually not very effective at the synaptic connections. So we needed to find something new. So we embarked on a kind of quest to find another light-sensitive inhibitor of synaptic activity, and we thought that the answer might lie not in this kind of microbial optogenetic or microbial rhodopsin light sensor, but actually in light sensors that come from other organisms or more complex organisms. We looked at rhodopsin proteins from various animals, and I'm not joking, these are the animals that we actually looked at. We mapped using genomic databases the types of rhodopsins that exist in these genomes. We cloned them, we put them into neurons. We went through many disappointments when many of them or most of them actually didn't do what we wanted them to do. But basically, eventually we managed to find a particular protein or a particular family of proteins, which is called bistable type 2 rhodopsins. I'm not sure if any of you heard about them, maybe some of you have, which if we put them into neurons, they actually traffic to the synaptic connections and shining light on these axons, now expressing this new type of opsin, which for the aficionados is a GPCR. Some people here know what this means. Actually allows us to do exactly that, to suppress the activity of synaptic connections and temporarily silence communication between two different brain regions. I'll just briefly show you how this works, because I'm almost out of time. So when you stimulate one neuron and record responses in another neuron, it looks oftentimes like this. So these are two synaptic responses driven by activity in an axon which activates synapses that release neurotransmitter. In this experiment, we expressed a mosquito rhodopsin, which is called OPN3. This mosquito rhodopsin was optimized to express well in mammalian neurons with several kind of genetic tricks. It allows us actually to shine light on the axon and almost completely suppress the release of neurotransmitters from these axons. And this is because this particular opsin actually is a GI-coupled GPCR, it couples to inhibitory signaling pathways inside the opsin, inside the neuron, and actually does what natural receptors that couple to these pathways actually do in the native neuron. So in that sense, it both works like a light receptor, but it also couples to endogenous signaling pathways, which makes it a very effective transducer of light into changes in neural activity. What you see here is the summary of synaptic responses at baseline, so in the dark, when this neuron is in the dark, and then after we shine green light for half a second on it, and you can see that the effect is very long-lasting because the opsin is actually extremely slow in reverting back to the off state. So a single pulse of light can actually suppress the synaptic activity for many minutes, which is useful for some types of experiments. We tested it in living mice. In this experiment, we expressed this mosquito rhodopsin in dopamine neurons and suppressed dopamine release into one side of the striatum, on one side of the brain. And as expected, what we saw is that mice expressing this rhodopsin actually showed a bias to start rotating in one direction because of the loss of dopamine signaling on one side of their brain, which is kind of similar to what we call a hemi-Parkinsonian state, in which there's a loss of dopamine neurons on one side of the brain. Now, why is this interesting? It's interesting because this is the first tool that allows robust silencing of synaptic transmission using an optogenetic approach. But it might also be interesting for translational approaches because we know that in many disorders of the brain, like Parkinson's, like epilepsy, there's a hyperactivity of certain parts of the brain, and suppressing activity could actually be a useful experimental approach to treating some of these brain disorders that are characterized by hyperactivity. Two examples of previous approaches that address this kind of question. One from the work of Jean Paz, who was also at Stanford at the time that I was there, and showed that silencing parts of the thalamus can actually interrupt ongoing epileptic seizures. So potentially a tool that allows us to do this at larger scale with higher light sensitivity might be useful to achieve this kind of suppression of epileptic seizures in larger brains. And another is that Parkinson's treatments are often utilizing deep brain stimulation. It's an electrical technique to rapidly stimulate axons in the living brain of Parkinson's patients. And in some patients that are irresponsive to medication, this actually saves them, but it also has many side effects. So an optogenetic approach to kind of recreating the effect with higher cellular specificity might actually be useful down the line in translation to humans. So I just gave you a taste of some of the questions that we're interested in, the tools that we've been developing. But just to summarize, our goal is to really understand the links between neural circuit activity and behavior and how neural circuit dysfunction leads to brain disorders. We've been developing optogenetic tools to allow refined control of specific brain circuits. And I showed you examples of excitation, inhibition, and projection-specific targeting with this mosquito rhodopsin that I was describing. And I think that from the work of Amiram, like much of the neurotechnology that has been developed in the past decades, you can clearly see how a new technique can actually open up windows into the brain that were not possible before. And I hope that some of the tools that we've been developing, we've been sharing them widely with hundreds of labs in the world, will actually lead to new kinds of windows that will allow new kinds of questions and hopefully also some answers. Just to thank the people who are doing the work at my lab, really fantastic team. I showed you work from many of them, but I won't go into the detail. Our funding, our collaborators, and thank you for staying all the way to the end.
So a technical question. So I fully understand how if you generate a knock-in mice, you can get super selectivity to an individual neuron or a few neurons using different promoters. But is it really so easy to do it using viruses? Because viruses, you know, it's difficult, I think, to make them as specific if you want to make conclusions which are based entirely on existing of inhibitor or stimulatory signal.
Yeah, that's definitely true. Viruses, the main limitation are that the viral particle is a 40 nanometer particle, at least the AAV one, and it's very hard to package a large genome into that particle. There's actually a very tight restriction on size. And what people have been doing is to try to find minimal promoter sequences that will allow kind of retain the specificity of the promoter without having the entire promoter sequence from the genome. And there are both promoter sequences and enhancer sequences that allow specificity to either excitatory or inhibitory neurons, for example, in cortex and hippocampus. There are also some minimal promoters that were developed for more unique types of neurons, for example, MCH cells in the hypothalamus, AGRP cells, orexin cells.
What percentage of the work are done entirely on using the knock-in when you know for sure? Or is it really now primarily using viruses?
So in the lab, when we work in mice, we sometimes use viruses in combination with Cree driver lines. So if you generate a Cree-driven virus, then you can inject it into any mouse strain, and the Cree recombinase will give you the specificity. The work with minimal promoters that are entirely encoded in the viruses has mainly been restricted to either excitatory or inhibitory neurons in cortex. In primates, actually, or in any other non-genetic animal model, you're restricted to using those promoters. And in that sense, but at least for excitation inhibition, this actually already works. There are many different promoters that have different kind of flavors.
So maybe a question from a non-expert who probably doesn't even know the right buzzwords to use. So for years I've been hearing about experiments where you knock things out. You knock out genes, in your case, you knock out connections. Maybe I missed something, but does it make sense to try to knock in connections between neurons and to see if that affects behavior? I mean, if the neurons are not connected, then they're not connected. And if they are, and you knock them out, you can tell what the role of that connection is. But if they're not connected and you connect them, maybe this has potential for making changes that will cause differences in behavior.
Yeah, that's a great question. It's a topic actually of a lot of neuroengineering type work. You can knock in connections in a way by applying plasticity protocols. So we know some rules of how neural plasticity works. So for example, if two neurons fire in very close sequence and they're connected to each other to begin with, that connection might be enhanced by the kind of co-firing, fire together, wire together kind of Hebbian plasticity. You can knock out connections by applying other plasticity protocols. So you can eliminate connections through long-term depression of synapses. It's very hard, I think, to generate connections de novo in the living brain.
You're answering about the techniques of how to do this, but I'm asking whether it makes sense. Say we can do this easily. Mm-hmm. Would it make sense to do this in terms of long-term future remedies or brain manipulation or whatever?
I think it does, and particularly some types of brain disorders or behavioral disorders have been proposed to result from kind of miswiring or maladaptive plasticity of circuits. For example, addiction, depression, things that are sensitive to all kinds of very crude manipulations like ECT, electrical convulsive therapy, which basically causes a seizure to the brain, somehow changes plasticity in the brain, which somehow helps depression patients. It's not really clear why, and it's a very crude approach. If you could pinpoint what's the actual circuit in which that kind of applying that kind of plasticity approach really gives rise to the effect of electroconvulsive therapy, that might actually be useful in treating that particular disorders, but there are many others.
A point which is important is when you knock in or you knock out, you can change the developmental program of a mouse. So if you do it at the embryos, you can really create mouse that has, that affected the nerve. So one has to devise a way to limit it without the developmental changes.
That's true, and actually when you use viral vectors, you can actually bypass this whole developmental thing.
This is the advantage of using viruses, but it has its own disadvantage.
That's true.
Yeah.
Mm-hmm. Okay, last question.
If you look at individual neurons, are they specific, one compared to the others, in what excites them, or what makes them work? Or is it only the position?
Yeah, so I think it was Ramon y Cajal that called neurons the butterflies of the soul, and he was saying that the neurons are so diverse that each one of them seems to be a unique thing. But he did actually, his work actually led to identification of neuron types which have different structure. We now know that different structure is linked to different physiology and also to different synaptic connections and network effects. So I would say that there are many types of neurons, probably dozens, if not hundreds, at least throughout the entire brain. But position and connectivity actually also define the role of a neuron in the circuit and not just its type or its gene expression pattern.
So where is the picture integrated?
That's what we're all interested in understanding, I think, in a way.
תודה רבה, עופר. תודה רבה.